So in this week's PCQ, we'll be going over the sliding filament theory, and more specifically, we'll be going over muscle contraction overview, structure of muscle cells, including fibers, myofibrils, and filaments, the key proteins, which are actin and myosin, and the sliding filament process, as well as the role of calcium in ATP. Okay, so muscle contraction overview. Muscle contraction is the activation of tension generating sites within muscle fibers, and the purpose of muscle contraction is to generate force, produce movement, maintain posture, and generate heat. This involves a series of steps where a motor neuron sends a signal, also known as an action potential, and this signal causes the release of acetylcholine at the neuromuscular junction. And then acetylcholine binds to receptors on the muscle fiber, leading to depolarization. This depolarization causes the sarcoplasmic reticulum to release calcium ions, and these calcium ions bind to troponin, which moves tropomyosin, exposing binding sites on actin. These myosin heads bind to these sites, forming cross bridges, and they also pull the actin filaments, causing the muscle fiber to shorten and contract. And then ATP binds to myosin, causing the cross bridges to detach. And then the cycle repeats as long as calcium and ATP are present. And then when the signal stops, calcium is pumped back into the sarcoplasmic reticulum and the muscle relaxes. This brings us to the structure of muscle cells. There are fibers, myofibrils, filaments, and this consists of thick filaments and thin filaments. So muscle fibers, this is what the entire cell is called, and it's long and cylindrical, and it contains multiple nuclei, and it consists of sarcolemma, which is the cell membrane of the muscle fiber, the sarcoplasm, which is the cytoplasm of the muscle fiber, and it contains organelles, and then the sarcoplasmic reticulum, and this is a network of tubules that store and release calcium ions, and then they are essential for muscle contraction. So there are three different types of muscle fibers. The type 1 is slow oxidative, which is efficient, fatigue resistant, and relies on aerobic respiration, and they're suited for endurance activities. Type 2, which is fast oxidative, these are faster and can use both aerobic and anaerobic metabolism. And then type 3, which is fast glycolytic, which are powerful but fatigue quickly, relying mainly on anaerobic glycolysis. Okay, so myofibrils, these are found inside each muscle fibers, and they are long cylindrical structures as well. They are responsible for muscle contraction and are made up of repeating units called sarcomeres. These sarcomeres are the basic functional unit of the muscle fiber, and they are defined by the region between two Z discs or Z lines, and they contain thick and thin filaments. Alright, so this brings us to filaments, and there's two types of filaments, thick filaments and thin filaments. The thick filaments are composed primarily of the protein myosin, and this myosin has a head that binds to actin, and then the thin filaments are composed primarily of the protein actin, along with tropomyosin and troponin. This tropomyosin and troponin regulate the interaction between actin and myosin. Okay, so throughout this process, there are a few key proteins, and the ones we're mainly going to be talking about today are actin and myosin. So the actin is the main component of thin filaments, and they have a binding site for myosin heads, and they form a helical structure. And then the myosin is the main component of thick filaments, and they have a head region that binds to actin and uses ATP for movement. The myosin head pivots, and this pulls the actin filament. So the sliding filament process, it's a pretty long process, and it contains multiple steps. The first being the nerve signal triggers release of calcium. So this is the start of muscle contraction, And this is where a motor neuron sends an electrical signal, also known as a nerve impulse, to the muscle fiber. This signal causes the sarcoplasmic reticulum, which is a network within the muscle fiber, to release calcium ions into the sarcoplasm. So the second step is the calcium exposes binding sites on actin. So in a relaxed muscle, the protein trypamyosin blocks the myosin binding sites on the actin filament, and when calcium ions are released, they bind to troponin, which is a protein complex attached to trypamyosin. And then the binding of calcium to troponin causes a conformational change in troponin, which in turn shifts trypamyosin away from the myosin binding sites on actin, and then this exposes the binding sites, allowing myosin to attach. The third step is that myosin binds to actin, also known as the cross bridge. Myosin is a motor protein, and it has heads that can bind to this actin, as we previously talked about. Once the binding sites on actin are exposed, the myosin heads attach to these sites, forming what we know as a cross bridge. The myosin head is already energized, having hydrolyzed ATP into ADP and inorganic phosphate, and is also known as it's in a cocked position, which it means it's ready to bind. Next is when the myosin pulls the actin, also known as the power stroke. And after forming the cross bridge, the myosin head pivots, pulling the actin filament toward the center of the sarcomere. This is the basic contractile unit of the muscle fiber, and this movement is called the power stroke, and during this, the ADP and PI are released from the myosin head. Next, we have myosin releases and resets with ATP. So after the power stroke, a new ATP molecule binds to the myosin head, and this binding causes the myosin head to detach from the actin filaments. The ATP is then hydrolyzed into ADP and PI by myosin ATPase, which provides the energy to recock the myosin head back into its high energy position. Now, the very last step is the cycle repeats, shortening the muscle. So if calcium is still present and the binding sites on actin are still exposed, the myosin head can bind to another site on the actin filament, and the cycle repeats. Each cycle pulls the actin filament a little further, causing the sarcomere to shorten. When many sarcomeres shorten simultaneously, the entire muscle contracts. This brings us to the last slide, which is where we're going to talk about the role of calcium and ATP. So calcium's role in a resting muscle, the protein trypamyosin blocks the myosin binding sites on actin filaments. And when a nerve impulse reaches the muscle fiber, it triggers the release of calcium from the sarcoplasmic reticulum. And these calcium ions bind to troponin, which is a protein complex on the actin filament. This binding causes a conformational change in troponin, which shifts trypamyosin away from the myosin binding sites on actin. This exposure allows myosin heads to bind to actin, forming cross bridges and initiating the contraction cycle. And then ATP also plays several key roles in muscle contraction, one being the myosin head activation. So ATP is hydrolyzed, which means it's broken down into ADP and inorganic phosphate by myosin ATPase. This hydrolysis energizes the myosin head, placing it in a cocked position, ready to bind to actin. Then the cross-bridge detachment. This is another one that ATP has a role in, and this is after the power stroke. ATP binds to the myosin head. This binding causes the myosin head to detach from the actin filament, and without this ATP, the myosin head remains bound to actin, resulting in rigor mortis. Myosin head reset is another one, and this is where the ATP is bound to the myosin head, and is then hydrolyzed, recocking the myosin head back into its high energy position, and it's ready for another cycle. And then the calcium pump is where ATP is also required for the active transport of calcium back into the sarcoplasmic reticulum. And this removal of calcium ions causes troponin to return to its original shape, allowing tropomyosin to block the myosin binding sites on actin, which leads to muscle relaxation. Thank you for listening to my little video on the sliding filament theory. And yeah, I hope you guys enjoyed.